Hello and welcome back everyone. My name is Shirsha and in this particular lecture series we are going to talk about one of the most commonly utilized methods for visualizing cells and molecules within the cells under microscopy, and that is immunofluorescence. So in this particular lecture series, we are going to have a couple of short videos in which we are going to talk about immunofluorescence and immunohistochemistry and cytochemistry. And finally, we are going to discuss about FACS or flow fluorescence assisted cell sorting. So before we dive into it, I would like to give you all a brief introduction on immunofluorescence. So what is fluorescence? Before we can understand immunofluorescence, let us brush up on what exactly do we mean by fluorescence. So fluorescent molecules absorb light of one particular wavelength, which is called the excitation light, and emit light of another wavelength, which is called the emission light. Now, how do they do that? So for that, let us take a look at the electron levels of a particular molecule and what happens when they absorb this particular excitation light. So electrons in any molecule or in any atom, let's get more precise. So in an atom, electrons exist in the ground state. Now the ground state might have multiple levels. So let us consider as 0 and your electron is residing over here. So when it absorbs the energy from light, that is photons, these electrons get excited, and upon excitation they will move from this state to the higher energy level, which is the S1 state. Now within the S1 state, your electron can move to either of these levels. It can go to either this 0 level or 1, 2, 3, or whichever of these high levels. Now once the electrons are in the higher level, they can either release some amount of energy and transit back to one of the immediate lower levels, like from 3 to 2 or 2 to 1, and when they transit back within this S1 level, then this energy that is released is very minuscule in amount, and this is called non-radiative transition. Why is it called non-radiative? Because no radiations are released post this transition. Now, when the electron has reached after this non-radiative transition from these states to this 0 state, it can jump back from here to the ground state. So it was in the excited state, now it comes back to the ground state. And when it comes back down to the ground state from this particular level, it will emit light. So essentially it emits energy, and because this energy is radiative in nature, so it will be emitted in the form of light, and what we now observe is called fluorescence. So this is your classical Jablonski diagram, which is used to explain fluorescence. Now, it is this phenomenon of fluorescence that we exploit in immunofluorescence. So, what exactly happens in immunofluorescence? The basics of immunofluorescence. So, in immunofluorescence, fluorescent compounds. So, if you think of the specific molecule that we were discussing about in the previous slide, whose Jablonski diagram we looked into, that compound is a fluorescent compound. So what is done is that such fluorescent compounds are usually conjugated to the Fc region of an antibody molecule. So what do I mean by the Fc region? So if you think of an antibody molecule, it has two heavy chains and two light chains. So on the top portion, if it's shaped like a Y, so how does an antibody typically look? So it has the heavy chains like this, and then there are the light chains over here. So essentially a Y shape. So within this Y shape, this particular part of it is referred to as the FAB portion or the Fab part, and this lower part is called the FC portion. Why is it C? C stands for, well, part of crystallization, but the FC portion is essentially constant within different antibodies derived from a same species that are of the same isotype. So C can also be considered to be synonymous to constant. So the fluorescent compound is usually conjugated to this Fc part of an antibody molecule, and because it is these Fab or FAB portions of the antibody molecule that are responsible for the specificity with which an antibody recognizes its specific antigen, so conjugation of the fluorescent compound to this antibody will not affect the specificity of the antibody, because your fluorescent compound that is depicted with a different color will be conjugated somewhere over here, and because the complementarity determining regions or CDRs are over here, so they can now recognize the specific epitope on the antigen that they were recognizing, and this fluorescent molecule over here will not really pose any steric hindrance to this recognition. So addition of the fluorescent molecule to the antibody does not impair its specificity or its ability to interact with the target antigen. So if antibody molecules are tagged with a fluorescent dye, which is called a fluorochrome, then the immune complexes that contain such fluorescently labeled antibodies can be detected by colored light emission when excited by light of the appropriate wavelength. So if we go back to the previous slide, whenever there was excitation, the electron was absorbing the energy from the photons and transiting from the S0 to the S1 state. And when it is coming down from the S1 to the back to the S0 stage, it is emitting light. Okay. So yes, it is very important to keep in mind that a fluorochrome can only be excited by light of a specific appropriate wavelength, because this wavelength will depend upon the energy difference between the S0 and the S1 states, and the wavelength of light, as we know, is related to its energy, the amount of energy that is stored in that particular wavelength. So this plays a crucial role in this. Now, antibody molecules bound to antigens in cells or tissue sections can therefore be now visualized. Now, the light that would be emitted by these fluorescent molecules will be of a different wavelength compared to the light that was being absorbed by it. Why? Because again, if you go back to the previous slide, in the when the fluorescent molecule electron was absorbing this energy from the excitation light, it could have moved from S0 to any of the S1 layers, that is level 1, 2, 3, anyone. But in order for it to come back to the S0 state, the electron has to be at the lowest energy level of the S1 state. And because it has already lost some energy in coming from the higher substates within S1 state to the lowest energy state within S1, so some energy has been lost in that transition in the form of non-radiative energies, right? So now it has reduced amount of energy. So when it comes back, or the electron jumps back from the S1 state to the S0 state, the energy that it will release in the form of fluorescence will be different from the amount of energy that it absorbed when it got excited, and therefore the emission light will be of a different wavelength as compared to the excitation light. Now, in order to detect fluorochrome labeled reagents, a fluorescence microscope is used. It is absolutely imperative requirement, in which the excitation radiation is transmitted through the objective lens onto the surface of the specimen. Absorbing radiation of appropriate wavelength causes the electrons of the fluorochrome to be raised to a higher energy level, as we saw in the Jablonski diagram. Now, as these electrons will return back to their ground state, light of a characteristic wavelength would be emitted. And this emitted light then forms a fluorescent image that we observe under the fluorescence microscope. Successful immunofluorescent techniques have a couple of requirements. What are these? Preservation of the antigen in a form that is recognizable by the antibody, because in immunofluorescence you will be visualizing molecules who are present within the cells. So these molecules must retain their integrity and their 3D structure, which will ensure that the specific epitopes that the antibody will recognize on them are maintained. So it is very important to ensure that the antigen conformation is maintained and it is in a form that can be recognized and bound by the antibody. Of course, you require a suitable antibody which is specific against your molecule of interest and an appropriate label. So next we are going to discuss about the different fluorochromes, and we'll be talking about some of the most commonly used fluorochromes. So each of the fluorochromes will absorb light at one wavelength and emits light at a longer wavelength. Now why will it emit light at a longer wavelength? So for this we have to go back to our formulas. So energy, or E, is given by the formula H C by lambda, where H is the Planck's constant, C is the velocity of light, and lambda is the wavelength. So as you can see from here, that energy is actually inversely proportional to the wavelength. So lower the energy, higher or longer would be the wavelength. And because when we are getting fluorescence, the energy emitted would be lesser compared to the energy which was absorbed by the molecule. So therefore, the light emitted would be of a longer wavelength. So let's clear this up. So one of the most commonly used dyes is fluorescein. It is an organic dye. It is the most widely used label for immunofluorescence procedures. It absorbs blue light at 490 nanometers wavelength approximately and emits an intense yellow-green fluorescence at a higher wavelength of 520 nanometers. So this is a example of cells which have been stained using fluorescein. To be more precise, it's an example of it's an image of smooth muscle cells stained with fluorescein. And as you can see, it emits a very intense greenish light. The next most commonly used dye is rhodamine. So it is also an organic dye. It absorbs in the yellow-green range of around 515 nanometers and emits a deep red fluorescence of 546 nanometers. Now, as it emits fluorescence at a longer wavelength than fluorescein, because fluorescein was emitting at the same range at which rhodamine actually absorbs light, so it can be used in two-color immunofluorescence assays. So what do we mean by two-color immunofluorescence assays? Say you want to visualize two molecules in the cell simultaneously. And why would you want to do that? So for instance, you want to see their co-localization. If say two proteins within the cell, A and B, are they co-localizing? That is, are they localizing in the same space, or is there any difference in their distribution within the cell? So for such purposes, you would typically go for a two-color immunofluorescence assay, wherein you can conjugate one of the dyes, say fluorescein, to the antibody against protein A, and the other dye, rhodamine, to another antibody which is specific against protein B. And this will now allow you to visualize both of them simultaneously within the cells. So this is an image of phalloidin conjugated rhodamine stained actin. So you can see the actin fibers which are present typically under the surface of the cell, under the surface of the cell membrane to be more precise. It's stained with rhodamine. So it emits a very, very bright red fluorescence. Phycoerythrin is another very commonly used fluorochrome which is used in immunofluorescence. So it is an efficient absorber of light, and in fact it is 30 times more efficient than fluorescein. So what do we mean by an efficient absorber of light? So efficiency of a fluorophore is essentially defined as a ratio of the number of photons emitted to the number of photons absorbed. That is, what fraction of the absorbed photons is the fluorophore emitting back out. So phycoerythrin is more efficient than fluorescein, and it also emits a brilliant red fluorescence. So these are some examples of phycoerythrin stained breast cancer cells. So you can see the cancer cells, they are roundish in shape, and you can see the phycoerythrin stain which is conjugated to the antibody specific against ER alpha. Now what is ER alpha? That is not really relevant to us, so we are not going to get into it. But yes, you can see what or how it actually looks like when visualized under the microscope. So it is important to keep in mind that fluorophores can lose their ability to fluoresce as they are illuminated. This process is called photobleaching. Now, why do fluorophores bleach or photobleach? This is because when you are continuously irradiating them with light of the excitation wavelength, it could lead to the destruction of certain covalent or non-covalent bonds within the fluorophore molecule itself, owing to the non-specific binding that can be caused by excitation light. And this would lead to breaking apart of the fluorophore molecule, albeit only transiently, and this will bleach the fluorophore molecules. So therefore, special care needs to be taken to prevent photobleaching through the use of more robust fluorophores that are less prone to bleaching. So in bleaching, you're essentially degrading the molecule by prolonged exposure to light of the excitation wavelength. So you have to take extra care when visualizing your cells or when imaging to ensure that your fluorophores do not get bleached, or you should go for using fluorophores that do not get bleached so easily. You can do this by how to avoid bleaching. You can increase the concentration of fluorophores, or you can reduce the intensity of light that you're exposing the cells to, or the duration for which you're exposing the cells to this fluorescent light. So next we're going to talk about fluorescence microscopes. So in order to visualize fluorescence, there exist special microscopes which are called fluorescence microscopes, and they are a type of light microscope itself. So what does a fluorescence microscope actually look like, and what are its components? So let us talk about that. So a fluorescence microscope is a microscope which is used to visualize substances using the phenomenon of fluorescence. Typical components of a fluorescence microscope include its light source, which could either be a xenon or mercury arc discharge lamp, the excitation filter, a dichroic mirror, and the emission filter. So let us now discuss what each of these components mean and what is their utility. Why are these components required and what function do each of them serve? So to begin with, let us talk about the excitation filter. The excitation filters are used to ensure that the specimen where you have your antibodies conjugated to the fluorophore bound to your protein of interest is irradiated only with light at the correct wavelength for excitation. So when your xenon or mercury arc discharge lamp produces light, it will emit light of all wavelengths. Now, you don't really require all of these rays of all of these wavelengths. Only light of a specific wavelength can excite your fluorophore molecule, and that should be enough and sufficient to serve the purpose. So the rest of the rays of the other wavelengths are filtered out using an excitation filter. Now, why at all go through this trouble? I mean, even if all of the wavelengths are irradiating your molecule of interest or your fluorophore, it will only absorb that specific wavelength which is required by, and it will simply ignore the rest of the wavelength. But then why do we filter out the rest of the wavelength? This is because when you will be visualizing your image, or when you'll be visualizing the fluorescence from these fluorophore molecules, you don't really want the excitation light, which contains light of all wavelengths, to mask the fluorescence that you will be getting or that would be emitted by the fluorophore. Now, why is it so? Because the fluorescence emitted by the fluorophore would be of a higher or longer wavelength than the excitation light. True, but if you do not filter out all of the other wavelengths from the excitation light, then the fluorescence would simply be masked by the excitation light. What do I mean? Let me clarify once more. So let us say your fluorophore is absorbing light of a wavelength X. and it is emitting light of another wavelength x plus y. Now because your excitation light, if you do not filter it out, also contains light of the wavelength x plus y, so you can't really distinguish that this x plus y wavelength light that you're getting, is it coming from the excitation light or is it coming from the fluorescent molecule itself. So therefore it is very important to have excitation filters. To help with the same purpose, there is an additional layer of protection which is called the emission filter, because like I already mentioned, the illumination light or the fluorescence that is emitted is much weaker, and the illumination light needs to be separated from this weaker emitted fluorescence light through an emission filter. The excitation filter was also separating or filtering out lights of these undesired wavelengths, but it is quite possible that some rays leak through and it is not 100% efficient. No thing is. So you have the emission filter present as an additional layer of protection to ensure that whatever you're observing is only fluorescence from the fluorophore and nothing else which is masking it. So emission filters only transmit light of the wavelength emitted by the fluorochrome, allowing images to be formed only by the emitted light. So this is what a typical fluorescence microscope looks like. In the next slide, we will take a look at a ray diagram and it will clear out the pathways taken by the light. But if you just briefly look at the fluorescence microscope over here, so this is the stage where you'll be keeping your sample. It can either be in different cell culture dishwares like 6-well plates or 24-well plates, or it can be on a glass slide also. So you have your eyepiece over here, and this is your objective lens which is placed just on top of the stage. So apparently it might look like an inverted microscope. We have talked about inverted microscopes in depth in one of my previous lectures on mammalian cell culture and the different techniques used in mammalian cell culture, but it is quite different. How so? In an inverted microscope, the source of light is typically on top. In an epifluorescence microscope, also the source of light is on top. But in an inverted microscope, the objective lens, that is this objective lens which will be detecting the or which will be collecting the image, is actually present underneath the stage, whereas in a fluorescence microscope the objective lens is actually placed on top of the stage, and this is one of the main critical points of difference. So let us forget, yes, the critical points of difference of a fluorescence microscope from an inverted microscope, and although it might look like an apparently an inverted microscope, it is not. So most fluorescent microscopes in use are epifluorescence microscopes. So I already mentioned this term in the previous slide and I will now elaborate on what it means. So in epifluorescence microscopes, the excitation and observation of the fluorescence, that is both excitation and observation, are from above, that is epi of the specimen. So if you take a look at this ray diagram, this will clear it up. So this is your light source, which can be your mercury arc lamp or your xenon lamp, and it is emitting light of all different wavelengths. You have this excitation filter over here. So the excitation filter will filter out the rays of all wavelengths other than your desired wavelength. And now this desired wavelength passes through. It faces a dichroic mirror here. Now we have not really talked about the dichroic mirror in the previous slide. What is a dichroic mirror? A dichroic mirror is a special kind of mirror which will reflect light of a particular wavelength, whereas it will allow light of a different wavelength to simply pass through it. It will not reflect it. So in this case, the dichroic mirror reflects the excitation light. So it will get reflected from here, it will pass through your objective lens, and here is your specimen on the stage, and it will excite the fluorophore molecules, and now there will be emission of fluorescence from these fluorophore molecules, which will make its way back through the objective lens, and now the dichroic mirror will not reflect it anymore. It will simply directly pass through it. There is an emission filter over here to ensure that any light that is leaking out from anywhere else, that gets filtered out, and now this light will be detected by the specific detector. So in fluorescence microscopes, dichroic mirrors play a very important role. So in this process, so unlike in other microscopic techniques where you transmit the excitatory light through the specimen, it is passed through the objective onto the specimen. So you don't really directly transmit your light through the specimen and then that will pass on directly through the objective. No, it is first passed through the objective and then onto the specimen. And since only reflected excitatory light filters through, all of the transmitted light is filtered out and it will give a much higher intensity. So it will ensure that only the fluorescence is detected, and because it is cancelling out the background, so now your signal will become much more intense. So here we have a very beautiful immunofluorescence image where you can see that three different stains have been used. So the blue stain used over here is actually called DAPI, that is used to stain the nuclei of the cells. And as you can see that we have one green fluorophore or fluorophore that emits light in the green range and another fluorophore which emits lights in the red range. So this is a classical example of visualizing two different molecules or proteins which have been stained using two different fluorophores. So this brings us to the end of this specific lecture. Over here, we just simply introduced the concept of immunofluorescence and what are the key components of a fluorescence microscope, how a fluorescence microscope works in very brief and just the very basics that is required from a biological point of view. As a user, the basic things that you should know when you're going to perform immunofluorescence. We talked about some of the dyes that are commonly available and that are commonly used, but let me reiterate over here that the list is exhausting and these are just some of the dyes that I talked to you about. There are more and more. Now there's an endless number of different dyes that you can use depending on your specific purpose, and the pair of dyes that you would select them such that their excitations and emissions do not overlap so that you can co-visualize in case you're going for co-visualization of multiple molecules. So in the next series or the next lecture of this particular series, to be correct, we are going to be talking about immunohistochemistry and immunocytochemistry. The two are very similar with very subtle differences in between. So we will be discussing about that. And finally, we will be moving on to talk about fluorescence assisted cell sorting. What is the basic principle of it and what is its utility and how is it utilized? So this was a very brief lecture and I hope I have been able to convey my point to you. In case you have any questions or any doubts, any queries, please feel free to leave it in the comment section and I will definitely get back to you with the answers. So that is it for now and I will see you again next time with the next lecture. If you like this particular video, do like the video, and if you are interested in more such content, do subscribe to my channel. Thank you and bye.